Incomplete dominance is when neither allele is completely dominant, and some of the F1 hybrids have a phenotype somewhere between those of the two parental varieties. In this case, there are 4 rabbits in the population. There are 2 males and 2 females. The minimum number of mating pairs is 2 to have a new population that still contains all 5 of the alleles. The gene C is found on a fur color locus on a chromosome. Gene C as a whole is one of the genes controlling rabbit coat color. The gene C has 5 alleles in which C is dominant of C3, then C2, then C1, and C0 is recessive. C is complete color for phenotype. Most of this population has complete color, and 1 is an albino. C3 is chinchilla, C2 is the allele for shaded, C1 is Himalayan, and C0 is albino. The first mating pair is C and C3 for the sperm or the male, and C and C2 for the female. When you cross this in a Punnett square, you get, for the genotype, 1 CC, 1 C3, 1 C2, and 1 C3C2. As a phenotype, 3 of these would be having dominant color or complete color, and 1 would be chinchilla. And this way, you have all the alleles that are being used are C, C3, and C2. The next mating pair is between C, C1 for the male, and C0, C0 for the female, which is albino. If you cross these, you get C, C0, C1, C0, C, C0, and C1, C0 for the genotypes. And for the phenotype, you have 2 with complete color and 2 that are Himalayan. This way, between both of these Punnett squares, all the alleles are being passed on to the next generation.